Topic of today's discussion is about the growth curve. This is of the bacterial growth curve. First of all, we will define how to define the growth curve. Very simple. You will tell that this is actually a kind of graphical representation in which we study the bacterial population, that how the population of bacteria is increasing with respect to time, or what is happening with the population of bacteria with respect to time. So how will you tell the definition? Simple. Graphical representation. Graphical representation of what? Of bacteria with respect to time. This is the simple, easiest definition. Okay, I hope you got. Now, what actually we study in this graphical representation? We study bacterial population with respect to certain phases like phase 1, 2, 3, 4. Now in the first phase, what is happening in this the bacterial population is not increasing. Only the metabolic activity of the bacteria are actually increasing. So what is happening here? By only bacterial metabolism is actually increased in this phase. No increase in the number of cells. In short, there is no cell division. Okay, only metabolic activity is seen in the lag phase. What happens when the time passes? So the population increases. How like? So the cell divides into two. One cell divides into two. So like this, that the second cell divides further into two. So like this, the population of the bacteria actually is increasing. So this phase where population is increasing, this phase is called as the log phase. So now oftentimes we feel confusion in the lag and log. So how to remove this confusion? Very simple. Consider log with a goal. So when you have a goal, a target, so you run for that. So this is how the bacteria run for the goal. That is, what is the goal of bacteria? The goal of the bacteria is to divide into two, means to increase its population. So it will be increasing its population, it will be running towards its goal. So the goal of bacteria is actually to increase the population, and that's why this goal is actually inversed as log. So log phase is actually the goal phase. Simply consider the log phase as a goal phase. Then you will never forget, or you will never be confused while you are asked the difference between the log and the lag phase. Okay, so this is the very difference. Lag in this phase actually there is no division. This is a kind of staying phase. So where here the division is seen because here bacteria is moving for its target, for its goal. So goal or log phase. And remember, log phase by the goal, you will never be confused then. Okay, so this is how the bacteria actually divide into two, and likewise the population is increasing with respect to time. As the time is increasing, population is increasing. Okay, this is x-axis and this is y-axis. On x-axis we have time, and on y we have the number of the bacteria that are actually produced. So in short, population is increased this way. This is called the log phase, where also called as the goal phase. Okay, I told you goal for the reason that you may never be confused. You may never confuse yourself while you are telling someone the difference between the log and the lag phase. Okay, lag phase, log phase. So log phase is here, the goal phase is here, and a point reaches when one bacteria is actually dying and one is actually formed or produced. So there is a kind of actually equal number of death and equal number of generation. So generation and death both is actually a kind equal. So here what happens? There is a kind standing phase seen. So that standing mechanism is actually called as stationary phase. The word stationary itself means standing. So here what is happening? What is actually seen hereby? Here the equilibrium is seen. So equilibrium is always considered as a kind standing. So there is no any increase or decrease in the number of population observed. What happens? The time passes. As the time passes, what happens? The number of this equilibrium shifts to one side. That is the death side. So the death then increase. So if one is produced, one bacterium is produced, in the meanwhile 10 will be actually degenerated, or you can say they will die. So now what is happening here? By 10 are moving towards the death, one is actually produced. So the death is more than the generation. So that's why this phase is actually shooting downward, and this is called as the death phase or decline phase. Decline means moving downward. So this is the downward phase called as the death phase. Why? Because bacteria are actually dying in this phase. So these are the four phases: lag, log, stationary, and standing phase, death phase. So lag is actually a kind of metabolizing phase. Log phase is the phase when the bacteria is dividing, and stationary phase is a standing phase, and the decline phase is actually the death phase. Why? Because a number of dying bacteria are actually more than the those that are produced. Okay, so I give you example that one is produced and 10 are actually dying in the meanwhile. So this is how we go through the growth curve, and still if you have confusion anywhere, you can ask us in the comment box, and thank you for watching.